Hello, I'm Flagg Flanagan, Chairman and CEO here at DiscGenics. This is a very exciting time in our business. We are currently conducting regulator-allowed human clinical trials in the United States and Japan for our first product candidate, IDCT, which is a homologous and allogeneic injectable disc cell therapy for degenerative disc disease. At the same time, we developed phase 1 manufacturing processes to support our current clinical efforts. Now, in preparation for an eventual commercial program, we are in the process of setting up our own large-scale manufacturing facility here in Salt Lake City. When this scale-up is complete, we will be able to generate thousands of doses of IDCT per lot to the highest global cell manufacturing standards.
My name is Laura Silverman. I'm the Senior Director of R&D here at DiscGenics. Today, we want to give you a glimpse into our research labs, where our team is working hard to improve and scale up the manufacturing process to generate IDCT in a GMP-compliant manner. We begin by procuring adult intervertebral disc tissue. The tissue is obtained from carefully selected donors and processed by our highly trained scientists. Cells derived from the disc tissue are grown in specialized conditions to generate the unique cell phenotype needed for IDCT to be active. The cells are being converted into the therapeutic progenitor cells that we noted were effective in animal models of disc degeneration. Here you can see individual cells growing and dividing into more cells over the course of 5 days. We're able to take measurements and counts of the cells to monitor growth and phenotype. Halfway through the process, we freeze the cells in the vapor phase of liquid nitrogen, which is negative 196 degrees centigrade. Freezing the cells gives us enough time to perform rigorous testing to ensure the safety and identity of the cells. After the cells pass the required testing, they are thawed and further expanded, this time using larger systems that allow for the generation of significantly more cells. During this time, we do in-process analytical testing to ensure the cells are growing properly. We also grow the cells in a bioreactor, currently in a small volume, but eventually in very large volumes in order to generate the many doses we anticipate we'll need to meet market demand. In this bioreactor, we are able to control variables such as temperature, pH, and oxygen in order to generate the optimal conditions for cell health, growth, and behavior. When the process is complete, we freeze the cells in their final form and perform release tests to evaluate the purity, potency, identity, and safety of our product. Using state-of-the-art equipment, we can evaluate these properties and ensure they meet preset criteria that we know is associated with clinical safety and efficacy for this homologous and allogeneic cell therapy. Each of these steps is critical to ensuring we are upholding the rigorous clinical safety and manufacturing consistency standards placed on us as developers of an IND-approved BLA pathway product. It is also what sets IDCT apart from minimally manipulated products that are available in the marketplace, yet have limited process controls and fewer analytical and clinical testing protocols. The final product is a disc cell therapy for degenerative disc disease, which is off the shelf. It's delivered to the patient via injection in a procedure room. As Flagg mentioned, we're currently executing regulator-allowed, double-blind, vehicle and placebo-controlled clinical studies in both the US and Japan. Once we demonstrate safety and efficacy of IDCT, we intend to approach the regulators with a biologic license application, which is a BLA in the US, and a conditional marketing authorization, which is a CMA in Japan. The steps we've taken to adhere to the many regulations of a manufactured cell therapy are plentiful, but we believe this is the best and only way to bring a product to the market that promotes patient safety and quality healthcare.
I hope you've enjoyed this look into our facility here at DiscGenics. We are very proud of the amazing hard-working team here in Utah, collaborating and innovating daily to bring this cell therapy to patients safely and effectively. IDCT has the potential to help millions of patients worldwide, and we are honored to be stewards of this game-changing medical innovation. As is our vision here at DiscGenics, imagine all the people we can help. So let me take you through, after this video, a little bit more about back pain that's experienced by a huge patient population worldwide, and tell you a little bit more about DiscGenics and our transformation from imagination to reality of delivering this drug to a major serious illness and a huge patient population. Low back pain affects approximately 1/4 of adults throughout their lifetime around the world. The spend in this country is more than $100 billion just in fusion surgery alone. It's the leading cause of disability worldwide. It's also the second biggest reason you visit a physician. And most importantly, it is the leading cause of non-cancer opioid prescriptions in the United States of America. So our product candidate is IDCT, injectable disc cell therapy. It is homologous and allogeneic. It is non-surgical, done in a procedure room. It takes about 10 to 20 minutes. It is a biomedically engineered progenitor cell. It's cryopreserved for off-the-shelf use, and it is regenerative and anti-inflammatory. Amazing technology. I'm so honored to be a part of this. We do show modes of action that's both regenerative and anti-inflammatory. We've done 24 different animal studies for our IND submission and our CTN submission, both in the US and Japan, where we've shown restoration of disc height and normalization of disc tissue and architecture that has not been seen in the controls. There's been no toxicity or safety issues associated with our animal trials, and we used all human cells in the animal trials to show the immunoprivilege of the cell itself. We did an athymic mouse study, and I want to talk about this study in a little bit more detail because we were asked by the agency from a toxicity and a tumorigenicity standpoint that they wanted us to do a live model, and we did that using the athymic mouse study, which is a very well-known model in various different modalities. We did that study, and not only did we not show any tumorigenicity, but we grew disc tissue. It was an amazing and exciting thing for us when we saw that technology and the ability for us to grow disc tissue in this athymic mouse model. And it was a very conclusive test that we did that helped us and really encouraged us going forward that we would have a successful product in clinical trials. So our process and our manufacturing process takes place in a two-step process. We procure disc tissue in an organ procurement. We process that in 2 steps, and we can bring forward many single lots of frozen product intermediate in this intermediate stage and develop just-in-time and accumulate product for the last part of the process for just-in-time processing. So as we build out our manufacturing and our dosing capabilities, we can use this 2-step process to build out inventory to be able to match the demand we need for the final cell product. We are currently in clinical evaluation in the US and Japan. US IND, BLA pathway, double-blind vehicle-controlled and placebo-controlled, multi-centered trial here in the US that will accumulate to 60 patients. We've already passed 2 safety reviews through 30 patients with the low-dose cohort. We have begun our high-dose cohort so far with no additional or adverse safety events. We feel really good about our pathway there. We have started in Japan enrollment there through a CTN with the PMDA. It is a double-blind sham control, and that study will accumulate to 38 patients, and we're really excited about that. We recently received a fast-track designation in this country. We're very excited about that fast-track designation because it will allow us to work more closely with regulators, hopefully to get an accelerated approval here in this country, and hopefully with the Japan accelerated approval there, we can get to CMA, or a conditional marketing approval. We think that's a very important step, and it really encourages us as we are moving forward with our manufacturing, as we talked about a great deal in the video, to scale out so that we can have cGMP doses ready by the time we can either get a conditional marketing approval in Japan or a possible accelerated approval in the US. So I've talked about our vision. Imagine all the people we can help. And you know, I wanted to put a slide up to talk about the growth that we've gone through to support that vision. You know, we were in University Park. We came there in 2012, and just recently we've moved to a 25,000 square foot facility, which will allow us the first initial build out of our doses, of our CGMP doses in Utah. We have the capability to expand that, and it's a very amazing place Providence led us into. So we're very excited about that facility, and you'll see a little bit more of that later, as you saw in the video. We started out with adherence to phase 1 GMP, and now we're scaling up to meet the CGMP standards. And it's a high bar, but we've got a tremendous team in place that will help us get to that point. We did doses at CTRM to support our US and clinical trials, and now we have a facility where it is our goal to eventually make 1 million doses a year. It's quite different from what Peter Marks talked about, and it's a very high bar, but we've got the team to make it happen so that we can serve this tremendous patient population that needs this product. We started out with 4 people doing scientific work in Utah. We've now scaled out to 27, and they're incredible people and incredible human assets. And then we have best-in-class consultants and support that really numbers, when I looked at it last night, really numbers almost 200 people that support us in various ways, from best-in-class consulting services to all the other services with those touch points. And we really appreciate all of those people that we've worked with and we're growing with to make this happen. Market size, you know, it's very big. And I think this is a very conservative size of the market. Literally millions of people have degenerative disc disease, and we think we can serve them very well. We also have a pipeline, and we're just not going to end with low back pain. We're already looking at cervical, which is a really tough malady that crops up. My wife had cervical disease. She had a 2-level fusion. She's now 8 years post-op with an adjacent level. I get encouragement daily to get this product to market. We're going down and looking at other things through the spine space to serve the patient through discectomy, adjacent level disease. And then we're also looking at possible articular cartilage and other things down the road, and even a second generation of IDCT. It is our mission to improve those debilitated by degenerative diseases of the spine through innovative regenerative therapies. And I will tell you that our core values, and we spent a lot of time putting these core values together of integrity, innovation, accuracy, and teamwork. And we really believe that through those core values, every single team member can run their business based on those core values and this mission. Here they are, an amazing team. And they walk this vision every single day in the imagination of all the people they can help. Thank you very much.